Block 2, we're going to talk about cell membranes, their composition, what their roles are, and cell signaling. Cell signaling is very important because that is what the body uses in order to carry out biological processes. So, but before we do that, just kind of a little recap on what we talked about yesterday. So, as we know, and this is we're talking about carbohydrate metabolism, we know we ingest polysaccharides that are broken down into monosaccharides. The main one being glucose. Glucose will then can either be, if there's too much glucose or we have enough glucose, it will be stored as glycogen via the process glycogenesis. If the blood is low on glucose and we need that glucose to rapidly increase, glycogenolysis will occur, which will break down glycogen and back into glucose. One of the primary pathways for us to attain energy using glucose is the glycolytic pathway. End product is pyruvate, which is converted to acetyl-CoA under aerobic conditions or when oxygen is present. Pyruvate is converted to lactate when oxygen is not present, and the Cori cycle converts that lactate back to pyruvate and glucose, and then it is returned to the muscle. Also, if there's too much glucose 6-phosphate, the glucose will that glucose 6-phosphate will be shunted via the hexomonophosphate pathway or the pentose phosphate pathway, with the primary end products being ribose and NADPH. Ribose is what? Used for what? Nucleic acid. And what is NADPH used for? Say again? Antioxidant. Yep, perfect. And then finally, pyruvate, if we need glucose and we don't have any more of it, there's a process called gluconeogenesis that will convert non-carbohydrate substrates back to glucose, which will travel to whatever tissue needs it to give us energy. Right? So, just some questions. What are some of the most important products of the hexomonophosphate pathway? Just said NADPH and ribose, right? Any questions about that? Yes, no? What does aldolase do?
It's an enzyme.
It's an enzyme, yes, definitely. One person tell me what it does loudly. Guys, I can't. Yes.
Splits fructose 1,6-bisphosphate into G3-DHAP.
Correct. It splits the 6-carbon sugar into 2 3-carbon sugars, right? Step 4, glycolysis. All right. One of you scholars, most of these questions I ask is because y'all scholars ask such brilliant questions. You have to make me go and look through research papers, which I thought I was done doing, but I still have to do it. But it's cool. Another question I got is, what happens to DHAP in glycolysis? That was a very good question. One person, somebody? What about the person that asked this question? What did I tell you?
It goes back to the other, I don't remember.
That's okay. DHAP
Like
goes
shuns
back to glyceraldehyde 3-phosphate. That was a very good question because I forgot to mention it yesterday. DHAP cannot be further oxidized, so it must be isomerized to glyceraldehyde 3-phosphate in order to continue on in the glycolytic pathway. Very good question. All right. And then question about glucose 6-phosphate dehydrogenase. It is X-linked recessive, so most common in males. Females can be carriers, and sometimes females do have G6PD, but it's very, very rare. And of course, I put a picture of the jumping jack. Maybe it's too early for that. Does pyruvate kinase deficiency, is it primarily found in children? No, it's found in both children and adults, and it depends on the disease severity. And of course, that severity will dictate what symptoms the infant or the adult has to varying degrees. Right? And the symptoms that we talked about yesterday, fatigue, shortness of breath, for example. Another very interesting comment I received from you guys yesterday was about lactate dehydrogenase deficiency. There are two types. One type is associated with muscle function, and that's the one where a person will have muscle intolerance and muscle wasting and breakdown. However, I believe the scholar that mentioned this yesterday, I believe your colleague or battle buddy had the LDHB because it doesn't affect the muscle, so that's why he was superhuman and was able to max out everything in the PT test. So, and this is just the different isoforms of this enzyme and where it's found in the body. And so this is the two types, and LDH muscle, LDHH heart, LDHM muscle, and then it's a tetramer, so it's a 4 subunit protein, and so basically it's just showing you that the different compositions that are possible and where they're found in the body. And so I'm pretty sure the person mentioned yesterday had LDHB because he didn't have any issues with his muscles. But he might be curious to know if he had any issues with like red blood cells or anything. This was something that I was not familiar with, so thank you for that question from yesterday. So now we're going to move into talking about today's lecture. That's cell membranes and membrane phospholipids. We know that the cell membrane is composed of bilayer membrane. Each layer has a head group that's polar or hydrophilic, and 2 fatty acid chains that are hydrophobic, and there are 2 sets of those. And so the structure of the membrane phospholipids are very important because they regulate what enters and exits the cell. They also, membrane phospholipids and cell membranes also are very important for cell signaling, cell communication, and also they contribute to a cell's shape and size and curvature, and that becomes very important. And we'll talk about some of this as we progress in biochemistry. So here's basically a diagram showing how cell membranes regulate the transport of material across its surface, and there are 3 different types. We're going to talk about these in a little bit more detail. Here is the cell membrane or lipid bilayer arrangement. You have aqueous phase, this is external, and aqueous phase internal, this is where the cytosol would be. And you see you have the hydrophilic head, hydrophobic tail regions, and then another hydrophilic head. This makes up the lipid bilayer. And there are also other molecules that are sometimes intercalated in between the cell membrane. For example, you have cholesterol, proteins, and sometimes even sugar moieties that are associated and intercalated in between the cell membrane, and they all have specific functions. And this makes up the fluid mosaic cell membrane model. And in this model, the cell membrane is fluid. It's not a fixed, rigid structure, but it's fluid. And these molecules that are either attached externally or internally or even integral to the cell membrane play a role in that function. So here are some of the most common membrane phospholipids. So all of our cell membranes are composed of many of these membrane phospholipids to varying degrees. We have cardiolipin, PG, or diphosphoglycerol, diphosphatidyl-glycerol, excuse me. And you have phosphatidylserine, PS, phosphatidylethanolamine, PE, phosphatidylcholine, PC, and phosphatidylinositol, PI. We're going to talk about each of these. We're going to talk about just some main characteristics of these and the roles they play in not only the cell membrane, but other roles they play in maintaining cell health and function. So these membrane phospholipids are integral to our cell membranes at varying degrees. So the most common membrane phospholipid, is phosphatidylcholine. That makes up about 40 to 50% of our cell membranes. And then cardiolipin is a unique one because that's found primarily on our platelet cells. We'll talk about that. So PG, cardiolipin, phosphatidylglycerol, all those names. This is a negatively charged phospholipid at physiological pH. This is very important in blood clot formation, and it is found in the inner mitochondria, and it basically helps our body maintain or it facilitates blood clot formations. And so people that do not have this protein or have issues with this protein have issues with blood clot formation. Phosphatidylserine is also negative at physiological pH, and it can be found intracellularly or extracellularly. It's also very important in nerve signal transduction, so it's involved in cognitive functioning, memory, and learning. And when it's found extracellularly, it's a signal to our body that that cell is toxic, so that will trigger our body to undergo apoptosis. It's where the cell is programmed to be died and to be removed from our body because it has some type of function that is not good for the body. Phosphatidylethanol, yes.
Sir, if we can go back onto the diphosphoglycerol.
Yes.
So you said that it helps with blood clotting, or does it decrease blood clots?
I'm sorry. So it's found on cells. It's found on blood platelets. So it helps decrease blood clots. Excuse me. So basically the role is that there are antibodies that produce. So how can I put this? There are people that have antibodies to cardiolipins. When they do, they have issue decreasing the blood clot, so they have blood clot buildup. And this is sometimes found. There's a particular test, and it's slipping my mind. I'll have the test tomorrow, but it's basically found during pregnancy. Bless you. Some women, their bodies are rejecting the fetus, and there's a test, a clinical lab test that's done that basically determines or detects this issue, and it's an issue with cardiolipin and autoimmune response to it. So if there's that issue, then basically the mother will spontaneously bleed out, and the fetus will not survive. So that's one way this is detected. It's commonly found in that. And I'll have more details about that test tomorrow. The actual test looks online. Any questions?
No, sir.
Any other questions? Okay. So phosphatidylanolamine, this is found in very low density lipoproteins, and this is LDL, which is the bad cholesterol. So this cholesterol carries triglycerides to our tissues. This is neutral at physiological pH, and it's primarily involved in the cell curvature because the ethanolamine is kind of a smaller head group. Phosphatidylcholine, it reduces the surface tension in lungs for efficient gas exchange, and so therefore it is important in pulmonary surfaction. And it's also involved to a lesser degree to anticoagulation properties with the protein C pathway. Phosphatidylinositol, PI. This is very important as a second messenger, and we're going to talk about this at the end of this lecture, being very involved in second messenger cascade systems. It's also involved, one of the forms of it is involved in taste and is associated with sodium channels, and so it's a salty taste, a gustatory channel. And this is negative at physiological pHs as well. Here is a summary of the membrane phospholipids that we discussed. Most important information, of course, is the name, the charge, and the primary functions of these membrane phospholipids. Any questions? Yes.
So when you use the abbreviation or like what you see as like a
It's probably going to be, if you see, it's probably going to be the full name, just the full name. Any other questions?
For the phosphatidylethanolamine,
Yes.
you have VLDL. What does VLDL stand for?
Very low density lipoprotein. So remember it's a cholesterol, LDL and HDL. So LDL is the bad cholesterol because it carries fats to tissues instead of removing them from the body. Any other questions? Yes.
So the PC and the PE, those are stable lipids?
That's a good question.
I see the ethanolamine and the
Yeah, I don't think they are, but that's a very good question. Those ethanolamine is definitely a amino alcohol, but I'm not sure if they're spindle lipids, but I will check back and let you know tomorrow. Very good question. Any other questions about that? Yes.
The structures for each of those membrane phospholipids?
No, that was just to add something besides words to the PowerPoint. You do not have to know. Not even a little bit. All right. So as I mentioned earlier, that there are in the cell membrane, there are molecules embedded in cell membrane that serve a certain function. Cholesterol is one of these molecules. Cholesterol helps maintain the optimal fluidity of the cell membrane. So it stabilizes the cell membrane at high temperatures, so the membrane won't become too fluid and allow molecules to just enter and exit at will. And at low temperatures, it's a fat. So at low temperatures, fats clump up and they get hard. And so at low temperatures, these cholesterols that are embedded and intercalated between the phospholipids will help the phospholipids not stick and come together. Does that make sense? in addition to cholesterol, there are also proteins that are either peripheral or integral to cell membranes. And proteins can make up a significant structural and functional amount of the cell membrane's surface. So you can have peripheral proteins located either extracellularly, shown here, or intracellularly. And then you have integral proteins, which span the entire cell membrane domain, and these integral proteins that span the entire cell membrane are typically their membrane channels, so they're pores, and they're involved in transport usually of molecules or information from outside to inside the cell. So there are three different types of transport across the cell membrane. You guys, I'm sure, are very familiar with these. The diffusion, facilitated transport, and active transport. Here is a picture of what's going on. I'm sure you guys know this. You guys have seen this many times, right? So diffusion, molecules go from high to low concentration. Facilitated, molecules go from high to low concentration using a transport protein. And then active transport, molecules go from low to high concentration using a transport protein and also energy, typically ATP. And this transport protein undergoes a conformational change during that process. Any questions about that? So these membrane proteins, or proteins that span the entire membranes, they often have secondary structures that we talked about, the alpha helices or the beta structures that we talked about in the last block. And so, of course, we know that since the inner region of the cell membrane is hydrophobic, these amino acids facing the inner membrane would be hydrophobic, of course. However, because either molecules or information is being transported across the cell membrane, the amino acids found within this channel are hydrophobic. Does that make sense? And the membrane channels, again, just like the different types of transport, you have simple channels. They have no control. They require no energy. It's just molecules going from high to low concentration. They can be selective for particular molecules. Aquaporins are an example of a simple channel. Aquaporins are found in the kidney and red blood cells, and they help the body maintain water homeostasis. Then you have facilitated transport proteins. These don't use energy, and molecules are going from higher to lower, higher to low concentrations across the cell membrane. An example of this is the GLUT1 transport system. So it's transporting glucose into red blood cells and the brain. And then active transport, we know, uses that carrier protein undergoing conformational change in order to get molecules across the membrane against their concentration gradient using energy. An example of an active transport channel is the sodium potassium ATPase pump, and this pump basically establishes sodium at high concentrations outside of the cell and potassium at high concentrations inside of the cell, and it also allows for nutrients and amino acids to pass through this transport system, and it keeps all of this is keeping the cell at the proper osmotic pressure required for proper cell function. So there are certain drugs that target membrane channels. And so in this case, they're membrane channel blockers. We're going to talk about 3 different types. Remember what I said about drugs, right? What did I say about it? Y'all don't remember? No? Drugs are bad. Well, in this case, they're good. But remember, if I mention drugs, then you will see them in this. Remember that? No? Well, if I mention drugs, you will see them. Okay. Does that make sense?
Specific drugs or drug types?
Say again?
Specific drugs or drug types?
Well, we're going to talk about specific drugs. So, first we're going to talk about calcium channel blockers. So, calcium channels are very important in smooth muscle contraction. What they do is these blockers basically decrease the transport of calcium into muscle cells. That decreases heart rate contraction, and then that will increase the diameter of the blood vessels, and therefore decrease blood pressure. So that's why we would treat somebody with high blood pressure with a calcium channel blocker. Amlodipine is an example. So if you see a drug with a dipine as a suffix, it is a calcium channel blocker. It's used to treat high blood pressure. All right. We also have 2 types of blockers that are used to help decrease acid formation. So if you have acid reflux, like I do, I have been given both of these drugs, and they are histamine receptor blockers and proton pump inhibitors, PPIs. So basically, these drugs both stop acid formation. Histamine receptor blockers stop parietal cells from releasing hydrochloric acid, and the proton pump inhibitors stops the final step of acid production. So if one of your patients were to have a serious acid condition such as Barrett's esophagitis, that condition basically so much acid is creeping up in the patient's digestive tract that his esophagus has now become highly acidic, and so that's very severe, very painful. It's going to cause a number of other conditions, and so this would be treated with the proton pump inhibitor, because a proton pump inhibitor is a longer lasting drug, and it's used for more severe acidic issues. Proton pump inhibitor would be omeprazole. So azole is a proton pump inhibitor. So if you have intermittent acid reflux, like after eating spicy foods or greasy foods, you might want to give your patient histamine 2 blocker, and that would be cimetidine. So any drug ending in idine would be used to treat like intermittent or sporadic acid reflux issues. Any questions?
Sir, is that just another name for Barrett, or is GERD more severe?
This is different from GERD. I think this is more severe than GERD. Yeah, because it's like, I don't think GERD makes your esophagus like, basically for this Barrett's conditions, the esophagus, let me put it this way. Barrett's disease is very severe GERD. So, because your esophagus in GERD, regular GERD, your esophagus, not all of it is necessarily eroded. Like I have GERD, but I don't have this severe GERD. I don't have any other side effects associated. There's a lot of number of other side effects associated with Barrett's esophagitis, including like internal bleeding and stuff like that. Any other questions? All right, now we're going to talk about
have
So-
a question.
Yes.
So these calcium channel blockers, can they be used when a female is pregnant?
I don't know, but I will find out. Any other questions? Right now, we're gonna switch gears and talk about cell signaling. So this is how messages are transferred or transported from outside of a cell into the internal portion of the cell. So there are 3 stages of cell signaling. We have reception, transduction, and response. Reception is the process in which a target cell detects a signal molecule from the outside of that cell, and the receptor protein located on the cell surface of that protein. So molecules detected on the outside of the cell. Transduction begins when that signal molecule attaches to the outside of the cell, and that starts the triggering of a chain reaction that leads to the response, which occurs when a transduced signal leads into a specific cellular response, biological activity. Typically, for example, we have like gene modification, and we're going to talk about some specific examples in this lecture. There are 4 different types of cell signaling: paracrine, autocrine, endocrine, and cell-to-cell contact. Paracrine signaling is a form of cell communication which cells nearby release signals, and then the signal gets to all of the cells in the neighboring area and cause a coordinated response of the cells. In autocrine signaling, basically a cell releases a molecule that then binds to that cell, and what happens is that the cell will then produce some type of signaling response or cellular activity. And so this is one way that cancer cells function, because they will release molecules that bind to cancer cells and also bind to neighboring cells. And that's one way that cancer cells propagate so quickly sometimes. And so autocrine signaling can also have paracrine components as well. Any questions? Endocrine signaling, we know that endocrine signaling is achieved by hormones. Hormones are made in one portion of the body and they're transported to another portion of the body. And there are several endocrine glands. I'm sure you guys have talked about many of them. And A&P, they include thyroid, adrenal gland, pituitary, hypothalamus, for example. And we have cell-to-cell contact, 2 types: gap junctions. There's small water-filled channels that directly connect cells together, so it allows for intracellular molecules to easily diffuse to other cells to signal a particular response. Then you have the cell-to-cell protein, receptor protein interactions, and in this signaling, cells with complementary surface proteins bind and transmit specific change, and that is particularly important in immune response. So this is how our immune cells recognize cells that should not be in the body, and they work to remove. And you have intracellular and cell surface signaling. So intracellular receptors are located in either the nucleus or the cytoplasm, and so molecules will pass through the cell membrane bind to the intracellular receptors in order to elicit a response, and that's what hormones do because hormones are lipophilic. Then you have, yes,
It's like our steroid hormones and stuff.
perfect, yep, absolutely, steroid hormones, and we're going to talk about that. And then you have cell surface receptors, and they bind, this is when a molecule or a ligand binds to an extracellular receptor, causing conformational change, and a second messenger system, which we talk about, will lead to a cascade of changes inside the cell. And so here is an example of intracellular and cell surface signaling. So you have intracellular receptor proteins, steroid hormones, for example. Steroid hormones are lipophilic, so they pass directly through the cell membrane. They bind to the intracellular receptors found in the cytoplasm and the nucleus. That will cause transcription factors to either replicate and transcribe DNA in order to translate it to messenger RNA and protein, and that will lead to a specific biological response. Here's just another example of how steroid receptors act. Again, they pass through the cell membrane with ease. They bind to the intracellular receptor that sends a signal to the nucleus to cause genes to be transcribed and translated into proteins, and that protein is going to exhibit a specific biological response. Any questions about that? That makes sense. Then we have cell surface signaling. So this is what happens when the receptor is found on the surface of the protein, and what happens is a ligand will bind to that surface receptor, causing a number of signaling cascades, either cyclic AMP, calcium increase, diacylglycerol, or tyrosine phosphorylation. We're going to talk about all these examples in biochemistry. These are all second messengers. Those second messengers will lead to a protein cascade that, again, can cause protein synthesis or can cause other protein modifications, and that will lead to a different biological response. Does that make sense? So one of the most understood cell surface proteins are G protein coupled receptors, These receptor systems are main targets for drugs because these receptor systems have a lot of functions in the body. And so how these proteins work is that they have three different subunits. There's conformational change. One of the subunits will interact with second messenger systems to elicit a particular biological response. And we're going to get into those details. So here is transmembrane G protein coupled receptors, GPCR. So these receptors have three internal subunits, alpha, beta, gamma. What happens when a ligand binds to the outside or the extracellular portion, that causes the alpha and beta gamma subunits to separate. The beta gamma form a dimer, and then that leads to a certain cascade of events and a particular biological response. There are 5 different classes of G protein coupled receptors. We're going to talk about G sub S, G sub I, and G sub Q.
Sir, what breaks the alpha and beta subunits?
It's broken when the ligand, the extracellular ligand, will bind to this protein. That triggers the conformational change of the protein. And then what happens is a phosphate interacts with this GDP to make it GTP, and then that triggers the cascade. And so here are the different types of G protein coupled receptors: G sub S, G sub I, and G sub Q. So G sub S stimulates cyclic AMP production via adenylyl cyclase and protein kinase A, and it's going to signal certain biological responses. G sub I inhibits cyclic AMP production, and then G sub Q stimulates phospholipase C. Now what I have here is all of the proteins. These would be the extracellular proteins: dopamine 1, histamine 2, vasopressin 2, that are associated with G sub S. And in parentheses is the ligand that binds to these proteins. So dopamine binds to dopamine 1, histamine binds to histamine 2, antidiuretic hormone binds to vasopressin 2. Same thing here. This is a muscarinic receptor. Acetylcholine is a ligand that binds to it extracellularly. Then we have adrenergic receptors, alpha 2, epinephrine, norepinephrine bind to it. And then same thing, histamine binds to histamine 1. Alpha 1, adrenergic, epinephrine, norepinephrine binds to it. And these highlighted mnemonics are basically the different proteins or receptor systems that are coupled to G sub Q. Have 1 M&M, G sub I, MAD2 because it's M2, A2, D2. And then just a quick acronym to help you remember what's associated with each of the different G coupled protein receptors. So we're going to go over an example of how this works. So again, the alpha subunit will detach from the beta and gamma, and when that occurs, the alpha subunit is activated when GDP is converted to GTP on the cytosolic side. That free alpha subunit will then interact with other membrane-bound proteins, for example, adenylyl cyclase, and that is going to cause signaling of second messenger molecules. So if we take the diagram we looked at before with the surface receptor protein, so if this is muscarinic 2 receptor, this would also be muscarinic 2. Remember, muscarinic 2 inhibits cyclic AMP production. So what happens is when acetylcholine binds to muscarinic 2 receptors, that's going to inhibit cyclic AMP production shown here and here. And in the case of this receptor system, that acetylcholine will bind to M2 receptors in the heart and cause heart rate to decrease. Does that make sense? Thank you, somebody. Are there any questions about this?
So the
Yes.
the receptor on the cell membrane is called M2. That's how you would reference that receptor.
Yes, it would be muscarinic 2 receptor. So all of these different receptors would be the surface receptors. And so these receptor systems stimulate phospholipase C. These inhibit cyclic AMP, and these stimulate cyclic AMP. All right, you guys take a 10-minute break.
What I said? Very chronological. It's like, geez, I know what we're talking about. I know that.
yesterday when we got to this slide. It's like, you just know everything, except for that type. And then I was just referencing last text and memorizing yeah. Or this text, I spent so much time on it. Just couldn't do it. A lot of vitamins. And I'm just like, that's right, more deficiencies.
I feel like you're, I think it's more like if you're not going to eat or jack and chunk, but if it's something that you step up and step back Oh, so I thought they did like a whole bunch of like 6.
Why they're not broken up?
Yeah.
I just think it's like
Okay.
so yeah, how do we do it?
I could actually do that quickly.
I don't think that would be a good question to know. If it's an inhibitory, and then like if it's inhibited, does that mean
No, I mean,
alpha
you won't
and beta gamma complex come back together and that's where it inhibits it?
Yeah.
Versus like you said if it's activated, the alpha subunit comes off and do this transduction and all these other interactions, but maybe the inhibitory end that subunits come back together.
Wait, go to the slide that has, it's like blue and it has G, I, GS.
Yeah.
So it's just put any
Like yeah, it could have been an A2 receptor, an alpha 2 receptor, or a
This makes me want to get like physically aggressive freaking Coast Guard.
yeah. But I'm curious. That's a good question. You should ask. Say subunits come back together.
You're gonna just vote word worse than like
Follow
how
that.
many times you beat out my brain. Fightable, but
Or keep it to yourself.
he doesn't teach any better.
You probably get to the gym. Imagine we have a whole lecture of micro. Like, after yeah. I was thinking like you might finish micro by like 11:30. And then you'll have like 11:30 and 3 I was thinking opposite. Or if you go on and I was thinking exercise. I'm like, takes me forever. I sweat long time.
I don't like it either.
That's why it doesn't.
Oh.
Yeah. So this is my wife's now second shift with sourdough. So you should have seen it last time. The last sourdough she made was half the height of this. Like there was nothing, like you couldn't do anything like it. So this one is like halfway there. Trying to get like the the big totally big, like fluffy bubble. I don't know how to Really?
Guess what? White matter.
But I'm talking like last time. I was like, golly, I hit it on the table. She's like, you're not allowed to eat my second. And I was like, hey, I'm not the one who's putting the work into it, but I'm saying I gotta eat it. I wanted to not break the chip too long. I was gonna buy really? Just put that on the end of my. I don't even need a rubber mallet. Is it I'll get sourdough. It's so hard. Electric cooler. You know?
Yeah, but it's like, I said which vegetable? I said which vegetable?
Oh, I don't know. That's a good question. I'll probably work on it today with all of our free time.
Let me refer back to my
Because I literally don't think anything else. I haven't even started my crew. Yeah, it's Mondays. It's only 6 days. You know, you start thinking about like that.
Pretty hard.
Yeah, that's good.
Oh, look, see this? That scores like each lap.
There's a lot of lists. The second one,
It says
get shit on them.
but it just reduces a little bit. Yeah, that's amazing. This
I do it. It's more pretty dumb.
if it's at all.
Yeah, if I had a PhD in microbiology, yeah, intro to biology for a bunch of dudes would probably be pretty
This
self-explanatory.
is this form.
I know.
Next hour, but Coast Guard.
Right now,
I feel like a show.
Pretty good. But we're gonna do a first overall.
Oh, wow.
slide deck baby.
Yeah.
If you'll rather have one,
It's not gonna happen. Apparently, like the whole like sometimes it's all going out the
it's an F word.
What's it like? Hang out with them. Like yeah.
Okay.
Yeah.
Any questions about how decoupled protein signaling occurs so far? Any questions about this pathway? I forgot to mention on this slide, so dopamine is the natural or endogenous ligand that binds to dopamine 1 receptors, as with histamine and antidiuretic hormones, vasopressin, and epinephrine and norepinephrine to beta 1. But these, dobutamine and isoproterenol, are drugs. And so these drugs bind to these receptors, so these drugs treat certain conditions. For example, what does albuterol treat? Asthma. So it binds to beta 2 that's going to cause vasodilation. Right? Does that make sense? Thank you. Any questions about that? All right, so just some more examples. So if this were the dopamine 1 receptor,
Wrapping up that summary on some membranes and signaling, the
dopamine, is that my co-lecturer? Dopamine, if this were a dopamine 1 receptor, that would be G sub S, right? So dopamine would bind to the external portion, triggering the separation of alpha from the beta gamma. Beta gamma will form a dimer. That alpha sub S subunit and GTP now would activate this adenyl cyclase, which will convert ATP to cyclic AMP, and also would trigger protein kinase A and a whole bunch of other different proteins in that cascade to exhibit a particular physiological effect. Right? Does that make sense? Now, conversely, we have alpha 2. This would be inhibitory. Either epinephrine or norepinephrine would bind, basically inhibiting the conversion, inhibiting the activation of adenyl cyclase, and thus inhibiting all the cascade events in this pathway to exhibit its biological effect. Does that make sense? Yes.
So if it's inhibiting that alpha subunit, is it going to have interaction with adenyl cyclase? That's the inhibiting part?
Adenyl cyclase will not be activated if it's inhibitory.
Okay.
So if it's excitatory or stimulatory, that alpha subunit will activate adenyl cyclase. Any questions about that? Now for G sub Q, what happens is G sub Q, for example, if we have vasopressin 1 as a receptor, antidiuretic hormone will bind to the external portion, and that will cause the alpha subunit to dissociate, the beta and gamma subunits will dimerize, and that will activate phospholipase C by the alpha Q subunit. This would be alpha Q, and that causes phosphoinositol bisphosphate to be broken down into DAG, diacylglycerol. Diacylglycerol chills out, hangs out in the cell membrane. Inositol triphosphate, IP3, leaves the cell membrane, goes into the cytosol, and causes the endoplasmic reticulum to release calcium. And calcium release will cause a cascade of events that leads to a specific physiological effect. Also, that DAG will activate protein kinase C while still in the cell membrane, that will cause a cascade of events that lead to a separate set of physiological effects. Any questions about that? I love the thumbs up. Thank you.
Well, for
Yes.
vasopressin specifically, that the calcium release, is that what's causing the restriction at the arterial level?
Yep. Yep. So that's the exact molecule that's causing the physiological. Great question. Any other questions? Right. So we talked about these different second messenger systems. For the stimulatory G couple protein receptors, you have adenyl cyclase. That's actually stimulatory and inhibitory. When stimulated, it will cause the conversion of ATP to cyclic AMP, modulation of protein kinase A. When G sub I, G couple proteins are activated, that adenyl cyclase will not be activated. causing certain physiological effects that will not occur. And then the diacylglycerol for the G sub Q receptor, G coupled protein receptors, you have the PIG2 is broken into DAG and IP3. IP3 leaves the cell membrane, goes into the cytosol, causes calcium to be released from the endoplasmic reticulum, and that causes a specific set of physiological effects. That DAG will interact with protein kinase C, causing specific physiological effects. Any questions? All right, here is breakdown of the adrenergic receptors and what their roles are. So we know epinephrine or adrenaline, it and dopamine increase heart rate, and this epinephrine ligand binds to alpha 1, alpha 2, beta 1, and beta 2 adrenergic receptors. Alpha 1 is G sub Q, which stimulates the phospholactate C pathway. Alpha 2 is G sub I, inhibits adenocyclase activation. And beta 1 and beta 2 are G sub S, and that stimulates the adenocyclase pathway. Are there any questions? All right, I will see you guys tomorrow, same time.
Thanks, sir. Thank you.